Finally, the last group of symptoms that I tend to associate with bacterial pathogens are the galls. Galls are basically undifferentiated masses of plant tissue that are caused by, in this particular case, infection by a bacterial pathogen. Certainly there are fungal galls and galls caused by other types of disease-causing organisms. There are also galls that are caused by non-diseases, so abiotic factors, but kind of the classic galls in my mind are caused by bacteria. The one that's shown here is something called crown gall. This is caused by a bacterium called Agrobacterium tumefaciens. This particular organism is oftentimes found in soil. It will infect roots and also oftentimes infects right at the base of the plant at the soil line, thus its name crown gall. And I've got a black arrow pointed to this large undifferentiated mass of plant tissue that's been induced by infection by this particular bacterium. What the organism does is it oftentimes gets in through wounds, and inside the bacterial cell, I've mentioned that there's a bacterial chromosome. This particular bacterium, in addition to that chromosome, also has a second piece of DNA. It's a circular piece of DNA called a plasmid, and there's a section of that DNA that actually gets cut out of that plasmid and injected into plant cells where it incorporates itself into the plant chromosomes. That particular little piece of bacterial DNA codes for a variety of proteins. Many of them are enzymes, and these enzymes help produce a group of compounds called the opines, and these opines are one of the favorite food sources for this particular bacterium. As part of all of this protein production from that little piece of DNA, you get this undifferentiated growth, lots of cells enlarge, and you get this kind of mass of plant growth that's produced, again, caused by this infection by the bacterium. Interestingly, scientists realized that what they could do would be to take that plasmid, cut out most of that group of genes that are involved in the formation of the galls, and then they can insert other types of plant genes or other genes in general into that little section of DNA, incorporate that DNA back into the plasmid, and then what they could do is stick the plasmid back into the Agrobacterium cell, have Agrobacterium do its normal infection process, and using this method, they could actually incorporate foreign genes into plants. The use of this bacterium was one of the first methods for genetically engineering plants.